Okay, good evening, and we are back here tonight to go ahead and move on to the lecture on the muscular system. So just as always, I'm just going to take just a moment here oops, I'm sorry, let me back up there. Take a moment here to go ahead and make my pen color something that you all can see. So I think we'll just go with yellow tonight. And again, so we are going to be doing this lecture on Chapter 8, which is the muscular system. So let's go ahead and get started. This is again I think a fun lecture because it's again some more basic hardcore anatomy. So we're really getting into some of the system lectures now and probably what you pictured you would be learning about when you took anatomy. So just for fun, we're going to go through some random muscle facts. These are really more for fun, not something that I'm necessarily interested in you memorizing for the exam, but it's a good way to start off the lecture. So just so you know, muscle, the word muscle actually means little mouse, and this was because of the way that the muscles ripple below the skin that, it looked like a little mouse. So that's how we got the name muscle. And just so you know, the smallest muscle, this is supposed to show, the smallest skeletal muscle in the body is called the stapedius, and that is the muscle that actually stabilizes the stapes, which was a tiny bone, one of your ossicles in your inner ear. And the largest muscle in the body is the gluteus maximus, hence the name Maximus. And there are about a total of about 650 skeletal muscles in the body. The most active muscle is the external eye muscles, which, as you can imagine, you're constantly moving your eyes. And then all of the body muscles contracting in one direction would give you about 25 tons of force. And my favorite muscle fact of the day, banging your head on a wall uses about 150 calories per hour. So those of you that, are feeling like that right now as we're in the middle of the material for exam 2, at least you're burning calories as you're banging your head on the wall. So, let's go ahead and move into the nitty-gritties in the lecture here. We're going to talk about some different muscle types tonight, so I'll actually come back to this slide a little bit later on. But we're going to talk about skeletal muscle, which is what this is here. We're going to talk about smooth muscle. Oops, I'm sorry about that. We're going to talk about smooth muscle, which is what this is here. Oops. And we are going to talk about cardiac muscle, which is what you see here. And by the end of tonight's lecture, you will actually be able to identify these 3 types of muscle based on some of the characteristics. You'll be able to look at a picture like this and know that this is skeletal, this is smooth, and this is cardiac. We're going to talk a little bit about the fact that all, obviously, our movements require muscles. And we're going to talk about how those muscles contract and how they use chemical energy in the form of ATP to create movements for us. Again, we introduced the 3 types of skeletal muscle that we'll talk about, and the focus on this chapter is mainly going to be on skeletal. So we'll talk a little bit about smooth and a little bit about cardiac, but mostly we're going to be talking about skeletal muscle. So let's get right into skeletal muscle and the structure of skeletal muscle. We're going to kind of break this down. Remember that, like when we talked about bone, each muscle here is going to be considered an organ. So, for example, if you're looking at the biceps muscle, the biceps brachia would be considered an organ, and that organ is made up of many different tissue types. So in this case, our skeletal muscles are going to be comprised of skeletal muscle tissue, go figure, right? And then On top of it, though, they also have connective tissues in them, and then nervous tissue for the nerves that are running through them, as well as blood and blood vessels. So again, remember that when you're talking about an entire skeletal muscle, that is considered an organ. It's made up of many different tissues. When we talk about the different types of connective tissue that you would find in a muscle, we'll talk about these connective tissue coverings. And so there's a layer of this really dense connective tissue called fascia, and that fascia actually will surround and separate each muscle. So this fascia is really going to be this outer layer that holds the muscle together, and then you're going to see it continues throughout the muscle and separates each muscle. And this fascia actually ends up at the end of the muscles extending out and becomes the tendons that actually fuse that muscle to the periosteum of the bone. So this fascia actually becomes the tendons. And again, that's what tendons, the purpose of a tendon, if you don't know, the purpose of a tendon is to connect a muscle to a bone. So we're going to talk about these three connective tissue coverings that make up our fascia, and that's going to be the epimysium, the perimysium, and the endomysium. Now, as you can see here, excuse me, the epimysium actually will be the connective tissue layer that covers the entire muscle. The perimysium is going to cover bundles inside that muscle, and the endomysium will actually cover each individual muscle cell. So I'm going to go ahead and draw this out for you. Because for most students, this tends to be the most challenging, one of the most challenging things for them to visualize. So when we look at a muscle as a whole, and I'm just going to draw, oops, excuse me, that was horrible. I'm going to start over here now. As you guys can tell, I'm not much of an artist. So you can have a good laugh at most of my drawings here. So here is my muscle. This is the entire muscle. And when we look at a muscle, if we were to take a cross section, remember taking a cross section would be like if you were cutting, a wire and then looking into it. So if we were to cut the muscle and look into it, this is what we would see. We would end up seeing that the muscle contains all of these bundles that we call fascicles. So this is a fascicle. And these fascicles are actually bundles of individual muscle cells. So I'm going to draw them in here. I'm going to draw them a little bit large just to make it easier to visualize what's happening inside your muscle here. So again, this is what each of these, and I'm not going to fill them all in, but each of these fascicles consists of individual muscle cells bundled together. And again, a little bit more terminology. Each individual muscle cell is actually referred to as a muscle fiber. The term muscle fiber means muscle cell. So I'm going to write muscle fiber, and then to remind you here, I'm just going to put in parentheses cell. So those are individual muscle cells. So now, the three layers of connective tissue that surround and kind of hold the muscle cells and the fascicles and the entire muscle together are going to be this. The first layer is going to be called the endomysium. So the endomysium is going to be this connective tissue layer that goes around each individual muscle cell. So that's endomysium, and then here is endomysium, and here is endomysium covering each individual muscle cell. So this is all endomysium. Each individual muscle cell has an endomysium surrounding it. It's right outside of what's called the sarcolemma, which we're going to get into in just a minute, but the sarcolemma is just a fancy name for the plasma membrane of a muscle cell. So for each of these individual muscle cells, we have endomysium, okay? That's what this is here, endomysium. And so again, it's just connective tissue that helps to hold those cells together. Now, okay, on top of it, here, these cells that are all bundled together are bundled together to form a fascicle. And what really holds them all together in a bundle is this layer out here that's going to surround. And this is, again, more connective tissue, right? Connective tissue doing what connective tissue does, and that is that it connects things together. So this is going to be called the paramycium because it goes around each of these fascicles. So there's paramycium, here's paramycium around here, and here would be paramycium around here. So all this stuff that you would see in between these fascicles would be paramycium. And then for our third layer of connective tissue, there would be a layer out here, okay? And this layer, excuse my horrible drawing here, but this layer out here, hopefully you can kind of get the picture here, and it would go again all the way around the cell, is going to be the epimycium. And that layer is going to be the outermost layer of connective tissue that is going to hold all of these fascicles, all of these bundles together. Okay, so those are the three layers of connective tissue. Again, we have the endomysium that goes around each individual cell. Then we have the paramycium, which is the connective tissue that goes around the fascicles. And then we have the epimycium, which is the connective tissue that surrounds the entire cell. So this is, again, diagram kind of depicting those different layers of connective tissue. So again, here you're going to see that, and this breaks it down into even what's inside an individual muscle cell, which we're going to get to in just a minute. But if you look here, this right here is a single muscle cell, okay? This whole thing. And I know it looks confusing because there's little bundles here. These are actually going to be your contractile bundles of contractile fibers inside the part that make up part of the cytoplasm of a single muscle cell. So here, if you see, look, this says muscle fiber, okay, or muscle cell. Again, remember that muscle fiber is the same thing as a muscle cell. It's just our terminology. And so this whole thing is a muscle cell. Okay. Now, if you look here, this connective tissue that runs right around here in between this muscle cell, and here's another muscle cell, here's another muscle cell, here's another muscle cell. So this connective tissue that's in between is the endomysium. Okay, that's your endomysium. Now, if you look here, all of these cells, okay, are bundled together in what's called a fascicle. So this, if you look right around here, this whole thing is a fascicle. Okay? And so that means that this connective tissue that's running around the fascicle right here is the paramycium. Okay? And then, if you look here, now this fascicle right here, okay, is just one of the fascicles that's bundled together to make up your entire muscle. So here's a fascicle, here's another fascicle, there's another fascicle. And so all of these fascicles are bundled together with this layer of connective tissue going around the outside called the epimycium. Okay? And that epimycium here covers the entire muscle and ends up becoming continuous along with the paramycium and the endomycium. They extend after the muscle is ended and become the tendon that connects then the actual muscle to the bone. So again, hopefully that will help between the drawing and this diagram. That will help you get a good picture in your mind of what's happening with these different connective tissue layers. Now let's look at an individual skeletal muscle cell. So remember again, and then when we say skeletal muscle fiber, that means that we're looking at a single cell. So it reminds you here, each muscle fiber is a single long cylindrical muscle cell. And now that I'm looking at this, I'm really sorry. The font on this is not a great font, so I won't use that again in the future because it just comes out looking really blurry. So again, if you have any questions on the slide, you can always email me. But so each of these muscle fibers, again, that's all it means. It's just terminology. A muscle fiber means a muscle cell. And muscle cells can be really long. There are some muscle cells that actually extend the entire length of a muscle. So again, even though they're these tiny microscopic things, they actually can extend the entire length of a muscle, so they can be quite long. When you actually look at a muscle cell, again, more terminology. There are a few terms that are a little bit different than what you're probably used to in terms of when we talked about cells in the cell lecture. One of them is that the plasma membrane of a muscle cell is actually called the sarcolemma. So the sarcolemma is actually the cell membrane or the plasma membrane of a muscle cell. And it's given a special name because it's going to have a slightly different structure in that it's going to have these little invaginations of the plasma membrane called the T-tubules. Literally where parts of the plasma membrane just pushes in, as you can see right here. You've got part that just pushes in and forms this what we call T-tubule, this little tubule that runs through the cell. And we'll talk about those in just a minute, why they're important. And then on the inside of the cell, instead of calling it the cytoplasm, we actually call it the sarcoplasm. And you can see everything is sarco. So in muscles, everything inside the cell is going to be, almost everything is going to be called sarco, sarcolemma, sarcoplasm, sarcoplasmic reticulum instead of endoplasmic reticulum. And again, we're going to talk about some of the different functions that these organelles do inside a muscle cell that's different than how they would typically function in another type of cell. So if we look inside a muscle cell, we have the sarcolemma again, which is just the plasma membrane, that outer layer of the muscle cell. Then we have these little invaginations of the sarcolemma called these T-tubules. And then I'm just going to go ahead and introduce this now because I'm going to be getting to it. If you look here, there's all of this stuff, all of these tubules that surround these T-tubules. So here would be the T-tubule right here. And then here, then there's all of this stuff here. And all of this stuff here is actually what we would consider to be endoplasmic reticulum in another cell, and here we're calling it the sarcoplasmic reticulum. And as you're going to see in a minute, it has a different function in that it's going to store calcium, and that'll be very important when we talk about sliding filament theory coming up here in a minute, and how that's involved with muscle contraction. So the other thing that you're going to see is you're going to have lots of mitochondria. Lots of mitochondria inside your skeletal muscle. And of course, the main function of that, mitochondria being our powerhouse of the cell, is that we have to have lots of ATP production for muscles to contract. They need ATP for muscles to contract. So we need lots of mitochondria. The other odd thing that you're going to see in skeletal muscle is that skeletal muscle are what we call multinucleate. That means that they have many nuclei. So again, it breaks kind of the rules that we talked about in the beginning of, a cell having one nucleus. There's exceptions to that rule, and definitely skeletal muscle is an exception to that rule. And it's one of the ways that you can identify skeletal muscle is that if you see a cell with many different nuclei, and they're pushed usually to the periphery, then that is a skeletal muscle. And if you look inside a muscle cell, the reason that the nuclei are pushed, and you can see that here, this would be a, oops, I'm sorry. This would be a skeletal muscle right here. I'm trying to draw on the lines there. There would be a skeletal muscle there. And you can see here, pushed out to the periphery are going to be the nuclei. So the nuclei are pushed away on the outside. And the reason that is that you have all of the contractile fibers in here. So here you can see these are the contractile fibers called your myofibrils. We're going to talk about those in just a minute. And so that's why you have the nuclei are pushed towards the outside. So myofibrils are going to be these bundles of contractile fibers. Bundles of contractile fibers. You can see that there. Contractile fibers. And we're going to talk about these contractile fibers here in the next slide. Sorry, maybe yellow was not a good color for this. So, again here, we're going to break down the inside of the muscle cell and get into some of the nitty-gritties as we look at these contractile fibers. So if we look here, again, this is a cross-section of a skeletal muscle, and if we were to take a closer look at these individual myofibrils, again, these myofibrils are these bundles. This is inside one single skeletal muscle cell. These bundles of what we call thick and thin filaments. These are our contractile fibers. The thick filaments are called myosin, and the thin filaments are called actin. And so what we're going to see is the actin and the myosin are going to be organized into these special little units, contractile units that we call sarcomeres. And a sarcomere is just a contractile functional unit of a muscle that consists of both myosin and actin. So what I want to do is I want to draw out a sarcomere for you so you can see this. Because you can see here the actin is these thin purple lines. That's the actin. And then this thick one here is going to be the myosin. And so what I want to show you is how the actin and myosin are organized together into these functional units that we call sarcomeres. So what I'm going to do is I'm going to go ahead and draw out a sarcomere. This is something that I suggest that you be able to do. If you can draw out this sarcomere from scratch and label all the parts, hint, hint, then this will help you tremendously on the exam. To be able to draw this from scratch means that you really understand the parts to a sarcomere. So what I'm going to do is go ahead and draw out a sarcomere for you. So here I'm going to draw as best as I can here my actin filaments. Oops, I'm sorry. My lines are a little wavy. I'm still getting used to my bamboo here. Okay, so these are going to be the actin filaments. And let's see if I can make them a little more even here. And then what you're going to see is you're going to have these fibers that run down here that, oops, I'm sorry about that, that are going to hold those actin filaments together. And then on either side you're going to see there are more actin filaments that are going to be part of the next sarcomere here. And they're going to be staggered off of those original actin filaments here. So now, hey, these are the thin filaments or the actin. Now we got to draw in the thick filament, which is going to be the myosin. So this is my thick filament here. Draw it in. That's going to be your myosin. And we'll talk about, oops, I keep hitting my button there, sorry about that. We'll talk about what that actin and myosin consists of in just a minute, or what these actin and myosin filaments look like in just a minute. So this is your thick filament. In between, that thick filament, again, is going to run in between the actin thin filaments. Okay? And then in this case here, we'll draw some thick filament. I'm not going to finish drawing the sarcomeres on either side because we really just need the one sarcomere in the middle here, but this will give you an idea of what it looks like when the sarcomeres are all joined together here. So now for some terminology. So again, here are these thin filaments. This is your actin. The thick filament here is going to be your myosin. And this line here is actually called the Z line. And the Z line is, again, it's just going to be this the special filament here that's going to connect or hold together the actin filaments. So one sarcomere or one functional contractile unit is going to be from Z line to Z line. So this entire thing here is a sarcomere. If we could draw that, this entire thing here is a sarcomere. Now we have what's called the A band, and I like to think of the A band as being the active band, if this helps you. It's called the A band. And the A band is going to run the length of the myosin. So it runs the length of the myosin or thick filament. Okay, and so this is your A band because, again, this is where the myosin is. Okay, so then you have, and I like to think of your A band as the active band. Again, hopefully that helps you in terms of remembering that, because as you're going to see in a minute, the myosin heads on this, the molecule myosin, are going to be what's going to creep along the actin and pull the actin filaments closer together. So again, they're really doing the action. Here now, the I band, here and here is going to be the band that consists of just thin filament. So this is called the I band. I band and I band. Okay. So again, this band here that consists of just thin filament or actin, I band is going to consist of actin only. So it has no overlap of myosin, no overlap with myosin there. So again, this entire band right here is the I band. This entire band here is the I band. Then we have what's called the H zone. The H zone is going to be in the middle here where we have just myosin. Okay, so you see all this here, there's no overlap with actin, it's just the myosin. So this is called the H zone. H zone. And so those are the major parts, the most important parts to understanding, again, a sarcomere and what happens with a sarcomere. And again, remember that a sarcomere runs, if I can draw up here, the sarcomere is going to run from Z line to Z line. That is the sarcomere. Okay? So those are the parts, again, to a sarcomere. We have Z line to Z line, and that equals the sarcomere. Again, we're in whatever, all the stuff in between. Then we have the H zone, which is just this area of just thick filaments. Then we have the A band, which is runs the length of the myosin molecule, and you have a little overlap with actin there. And then the I band is going to be just the actin filaments here. Now this is important to understand because when we talk about sliding filament theory in a minute, we're going to talk about when the myosin heads creep along and pull these two actin filaments together. And as they pull these actin filaments together, our H zone is going to get smaller, and then our I bands are going to get smaller. But it's going to be important to remember that the A band will not change in size. It's only the I band and the H zones that will get smaller during muscle contraction. So real quick, I'm going to show you an animation here, possibly. I may have to turn my pointer off here. Oh, there we go. Let's see. And real quick, I'm just going to go to the arrow. I'm going to have to flip back and forth because I have a couple animations here that I think are really helpful in terms of understanding muscle contraction. Muscle contraction tends to be, again, I think more of a visual, it should be more of a visual lecture because it really helps to be able to kind of visualize what's happening, these individual parts inside a muscle. So here I'm going to show you a little animation of a sarcomere and what it looks like when a sarcomere shortens. So let me expand that out here, and you'll actually get some audio with this as well, which is nice.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament.
So you can see the H
As a result, the
zone
sarcomere is
and the
shortened.
I band decreasing.
In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
So again, this is a really nice animation of a sarcomere shortening, and I actually am going to post these animations for you guys because I think they're really helpful. So that's why it's important to understand the sarcomere so you can see what happens as the sarcomere shortens.
In a relaxed muscle, actin
here,
and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact.
Creep along
the actins slide toward the center of each myosin myofilament.
And as, again, those myosin heads creep along the actin, you're going to see, again, that the I band decreases and the H zone decreases as well. But again, that A band is not going to change in size. It's going to stay the same. Okay? And your overall sarcomere will shorten as well from Z line to Z line. So again, hopefully that'll help you in terms of visualizing what we call sliding filament theory, which is what we're going to get into coming up here. So let me go ahead and back out of this and go ahead and get back into our lecture here. Sorry about that. So, and I'm just going to swap back so that I can use my pen again. So excuse me for a second there. Okay, so that's the sarcomere. And actually, if you look back here, that's what this is a picture of. So this is showing you, it's basically trying to show you the same thing that I just drew. So you can see Z line to Z line here is one sarcomere. In purple here going all the way across would be the myosin, and that makes up the A band. And the H zone would be just myosin heads. Here, the dark purple is showing the myosin and the actin overlap. And the I band here would be, again, just the actin. So hopefully that helps you in terms of visualizing, again, what a sarcomere is. And so if you look inside a myofibril, you have many sarcomeres, many of these contractile units that are again, you know, right next to each other. They're crammed right one next to the other, next to the other. And so we'll talk about this is how a muscle cell is going to contract when all of these myofibrils are contracting. Okay, this is kind of cool because this is actually what's called a transmission electron micrograph image of a sarcomere. So this is cool because as you can see, this is actually Z line to Z line here. And here would be your A band right here. This is your A band. So that would be your A band. This, again, being Z line here. I know you can't see this very well. My yellow on here, but and then here in the middle would be the H zone, somewhere in here, and then here, from here on out would be your I band. So this would be your I band. Okay, so again, this is just going to show you now an overview of an entire muscle cell, and I think it's a nice picture to visualize again what a muscle cell looks like. So you can see here are your myofibrils, and you can see again your myofibrils consist of all of these sarcomeres, these contractile functional units. So here's a sarcomere, and there's a sarcomere, and again those sarcomeres you'll find on individual myofibrils. Well, then you find many myofibrils inside a single muscle cell. And then you can see you've got your nucleus here on the periphery. And then you have lots of mitochondria. And then again, as a reminder, remember that you're going to find that you have the sarcolemma on the outside here. The sarcolemma is just the plasma membrane of a muscle cell. And then you have these little invaginations of the sarcolemma called T-tubules that are running down here. And then you have all this stuff that surrounds, all this stuff here, that pink stuff there, that's got all these tubules, this network of tubules running across here. All of that is called the sarcoplasmic reticulum. So again, keep that in mind, keep that structure in mind of what that looks like inside a muscle cell, because we're going to get to the importance of the T-tubules and the sarcoplasmic reticulum coming up real soon here. So now let's talk about what creates the stimulus for a muscle cell to contract. And then after we do that, then we'll actually get into how that muscle cell contracts and what's happening inside the muscle cell. So this is called a neuromuscular junction, and it's just like it sounds. It is a junction between a nerve and a muscle. And obviously the nerves, as you probably already know, your nerves are what are going to ultimately tell a muscle whether or not to contract. So again, you have to have these neuromuscular junctions for the nervous system to control muscle contraction. So the site where your motor neuron, and so this is called a motor neuron. A motor neuron is going to be a neuron that's going to innervate or connect with a muscle or an organ. And in this case, obviously, we're connecting with a muscle, so that's hence the name neuromuscular junction. So you can see here is your motor neuron connecting with the muscle cell here. So now when you look at a motor neuron at the very end of it, your motor neuron is going to branch into these things that we call axon terminals. And you can see that they'll form many axon terminals. So they'll actually touch the muscle cell in multiple places. Now, when we look at the area where a motor neuron actually touches the muscle cell, you're going to see that the sarcolemma of the muscle cell is all folded here. Okay? And this is what we're going to call the motor end plate. Motor end plate. So put it off to the side here. Motor end plate. And that motor end plate is just the area that where we have a neuromuscular junction where the sarcolemma is folded. And the reason it's going to be folded is that folding, and as you're going to see, this is going to be kind of a theme that we're going to see throughout anatomy. Anytime that you see a structure that's folded, typically the purpose is to increase surface area, to maximize surface area. So in this case here, we want to maximize surface area so that when this neuron here releases its neurotransmitter, which it's going to, into this little gap here, that there's lots of surface area with lots of receptors on it to be able to bind to that neurotransmitter. So again, we're going to get into, don't worry too much if you didn't get all that, because we're going to get into the nitty-gritties on this in just a minute. So when we look at our neuromuscular junctions, then we can branch kind of, so we can, if we can go back out and move it back out, we're going to talk about what's called a motor unit. And a motor unit, it consists of a single motor neuron and all of the muscle fibers, all of the muscle fibers that it controls. And what this means is that one single motor neuron will reach out and oftentimes touch many multiple cells. So it'll branch just like it did here when we saw, again, you've got it, the motor neuron branching. It'll branch, but it oftentimes branches and then touches many multiple muscle cells, or controls many multiple muscle cells. So oftentimes you're going to see that one motor neuron will control many muscle cells. So that's what we're talking about when we refer to a motor unit. A motor unit equals a motor neuron and all the muscle cells. Again, muscle fibers, same thing as muscle cell. All of the muscle fibers that it controls. So again, that's really important to remember that terminology, because as we go on, we're going to talk about something called motor unit recruitment. So here again is a closer look of that motor neuron, and you can see this is a branch coming off of the motor neuron, and here these are called the axon terminals. And again, these axon terminals of your motor neuron are what are going to, if you want to say, join up with the muscle cell. And the muscle cell, in those areas where they're meeting up with the axon terminals, again, the sarcolemma, the cell membrane there, is going to be folded. And that is what we call, again, the motor end plate. The folded sarcolemma equals the motor end plate. Okay, so this is kind of cool because this is actually, again, just a microscope image of neuromuscular junctions. So this is cool. You can see here, all of this is the motor neuron here, and you can see that, check this out, these are all of these branches coming off of the motor neuron. There's an axon terminal, and here would be another axon terminal here, and you can see this right here is one single muscle cell. This is another muscle cell. This is another muscle cell. So if you look, this one motor neuron is controlling all of these muscle cells. This is what we would call a motor unit. Okay? That single motor neuron and all of the muscle cells that it connects to. This is just kind of a neat picture. It shows you, again, a microscope image, close-up now, of each of these neuromuscular junctions. So here is, again, where that motor neuron has branched. Here's an axon terminal that's going to control this muscle cell. Here's one that's going to control this muscle cell. Here's one that's going to control this muscle cell, and so on. And that's just a close-up again of, we'd be looking at here, this is the axon terminal of your motor neuron, and then it's stained dark because you're also going to have the folded membrane, the folds in that skeletal muscle membrane that are called the motor end plate. And this is a scanning electron micrograph of your neuromuscular junctions. So here you can see here would be a neuromuscular junction, and here's a neuromuscular junction. So now let's talk a little bit about the process of skeletal muscle contraction. So when we talk about muscle contraction, it's actually going to involve many different components that will ultimately result in the shortening of sarcomeres, as we've kind of already talked about, and the pulling of the muscle against its attachments. So again, for the most part, its attachment is bone. So now let's take a closer look at the actin and myosin that makes up the sarcomeres. So if you look, the myosin, or the thick filament here, so here's your myosin filament, right? Your myosin consists of these two twisted strands with globular cross bridges. These are the globular cross bridges that are sticking up here and here, that project out along these strands. And so if you could picture, if you had, the way that I always pictured myosin would be if you had golf clubs that were, you have one golf club here and another one here, and they are connected and twisted together in the center. So you have, your golf club head here and your golf club head here that make up the cross bridges. So if you could put, again, golf clubs end on end with each other and twist them, then that would be kind of the best image that I could picture of your myosin molecule. And of course, you have many golf clubs here, just like you have many myosin molecules end to end that are twisted together. So this is your myosin molecule, and you have all of these myosin cross bridges, or what we're going to call the myosin heads, that are projecting up here, all these myosin heads. And then actin, so this is going to be your actin, okay? Your actin filament kind of looks like you've taken a pearl necklace and you've twisted it. Okay? And so actin is a globular protein, and it has these myosin binding sites on them. So if you look here, you can barely see them. They're in purple here. These are the myosin binding sites, and they're covered in this picture here, because this would be in a relaxed state. But when a muscle is in a contracted state, those myosin binding sites are exposed, and that is where myosin is actually going to bind with actin temporarily. The other thing that you're going to see in your actin filaments is you're going to see this strand called tropomyosin here. So you see these strands called tropomyosin. And you're going to see these little molecules called troponin as well. And troponin actually interacts with the tropomyosin. So we oftentimes call it the troponin-tropomyosin complex. And so we're going to talk about the importance of the troponin-tropomyosin in just a minute here. But you can see from this diagram, one of the main important things is that right now it's covering that myosin binding actin, or that myosin binding site on the actin. So again, this is kind of just a better picture of showing you a single myosin molecule, and then when you put a bunch of these myosin molecules together, and then end to end and twist them together in the center, that's what your thick filament looks like. And you can see all these myosin heads sticking up from it. And then again here, this is just showing you an actin filament. And this is a better picture because now you can see here's the troponin molecule, and then here is the tropomyosin that it interacts with. Those strands are the tropomyosin. Okay, and then just to remind you again, this is what you're going to see in the sarcomere. You're going to see again the thick filament that consists of the myosin, and then you're going to see again on either end here, you're going to have these thin filaments that's going to consist of the actin, the tropomyosin, and the troponin. Okay, so this is where we get into the good stuff. So now we're going to talk about all of this and how all of this leads to ultimately muscle contraction. And so there is a theory on how muscle contraction occurs, and it's called the sliding filament theory. Now, please keep in mind that when we use the word theory in science, theory means something very different in science than it means out there in the general public. In science, a theory is something that has been so extensively tested that we consider it to be fact, scientific fact. So, sliding filament theory is, because it's been so extensively tested, what we accept for as scientific fact the process by which muscle contraction occurs. So, according to sliding filament theory on muscle contraction, the myosin cross bridge here attaches to the binding sites on the actin filament and bends. So those myosin heads are going to undergo what we call a power stroke, and they're going to pull on the actin filaments. And then it's going to release and then attach to the next binding site, pulling on the actin filaments even more. And so what's going to happen is you have these myosin heads sticking up here from this thick filament. And what they're going to do is they're going to pull, they're going to creep along these actin filaments, pulling the actin filaments closer together. So again, on this side, the myosin heads are going to bend and creep along this actin filament here, pulling it this way. And then here, these myosin heads are going to pull this actin filament in this direction. Okay? Ultimately shortening that sarcomere. Now we're going to talk about how they do this, and one of the main ways that they're able to do this is they're going to use the energy from converting ATP, our energy currency of the cell, to ADP, and they're going to use that energy to cock the myosin heads so that they'll be ready to undergo that power stroke and creep along the actin filament. Now this is kind of important to understand because ATP, if you remember, ATP refers to adenosine triphosphate, which means it has three phosphate groups. That's supposed to be a P there. 3 phosphate groups. That's ATP, adenosine triphosphate. Now what you need to just kind of keep in mind is that these bonds, these phosphate bonds here, are high energy bonds. And so if we were to break this bond here, which is what's going to happen in the process of muscle contraction, that if we were to break that bond, what we're left with is adenosine diphosphate. Diphosphate means 2. So that would mean that you'd be left with adenosine plus your 2 phosphates and then an individual phosphate group right here that was cleaved off here. So the reason that we would do this is the energy from this high energy phosphate bond can then be used to cock the myosin heads. Okay? So that's how ATP is going to be used as a form of energy to cock the myosin heads. Now I have to give you again, I'm kind of giving you the pieces to the puzzle, and then we're going to put all this together. So now we're going to go back and talk about how this process starts, and then we'll get back into the nitty-gritties on what's happening in those sarcomeres. So when we look at the very beginning of muscle contraction, it has to start with this motor neuron. What's going to happen is the motor neuron is going to release a neurotransmitter called acetylcholine. So again, this is the nerve, the motor nerve. It's going to release something called acetylcholine. Now, acetylcholine is the neurotransmitter. It's released into what's called the synaptic cleft. And the synaptic cleft is just going to be the space in between the motor neuron and then the muscle cell itself. And so what is going to happen is if you were to look at, if you remember the motor end plate, or again, this is just the sarcolemma, that cell membrane, the sarcolemma of the muscle cell. And remember, it's folded where it meets up with the neuron, where it meets up with the nerve, the neuron. So what it's going to look like is if you were to take a close-up look of that motor end plate. Okay? You're going to see that it's studded with all of these receptors. And so that's what you're seeing here in blue and red. These are receptors. And those are receptors for neurotransmitter. So there's tons of them. And so as we release neurotransmitter, so as the motor neuron releases neurotransmitter, and in this case it's going to release a very special neurotransmitter called acetylcholine. That acetylcholine will bind to the receptors on this motor end plate. So these balls here are supposed to simulate the acetylcholine binding to the receptors on the motor end plate. So when that happens, when these receptors on the muscle cell bind acetylcholine, okay, that is now going to stimulate a muscle cell impulse, or what we're going to call an action potential, to travel across the muscle cell. And it's going to spread over the surface of the sarcolemma and into the T-tubules, where it will finally reach the sarcoplasmic reticulum. Now remember, the sarcoplasmic reticulum surrounds those T-tubules. So this is again just what a neuromuscular junction looks like. So again, as we get a signal in the form of an action potential that travels down our motor neuron, it's going to signal this motor neuron to release acetylcholine. So you can see here, these are vesicles that are just storing acetylcholine. And so when it gets the signal, those vesicles are going to fuse here at the end and release, they're going to fuse with the membrane of the motor neuron here and release the acetylcholine into what's called the synaptic cleft. This is the synaptic cleft. So here you can see all of the acetylcholine that's traveling across in red dots here. That's the acetylcholine traveling across that synaptic cleft. And then that acetylcholine will bind to receptors on the surface of the muscle cell. Here are those receptors, the acetylcholine binding to those receptors. And so again, they're binding to the receptors on the motor end plate. That's this folded area of the muscle cell. And remember, this motor end plate has tons of these receptors here that are going to bind acetylcholine. And as soon as they bind that acetylcholine, boom, now it's going to pass the message onto the muscle cell, meaning that now we're going to stimulate, just by binding that acetylcholine, we're going to stimulate an action potential or a muscle impulse in the muscle cell. So again, upon receipt of a muscle impulse. So again, as soon as that muscle cell has bound acetylcholine and that acetylcholine is stimulated an action potential in the muscle cell, that impulse is going to travel down the T-tubules, and as it travels across the sarcolemma and down the T-tubules, it's now going to stimulate the release of calcium from the sarcoplasmic reticulum. So that's the main function of the sarcoplasmic reticulum in muscle cells, is to store calcium. And so now we're going to release calcium from that sarcoplasmic reticulum. Now that calcium, and this is where again you may have to watch this lecture a couple times because I know it's involved, and so you may have to write this out step by step for yourself because there's lots of steps in this process. But now that calcium that's released from the sarcoplasmic reticulum, okay, is now going to go and it'll be released into the sarcoplasm, basically into the cytoplasm that's surrounding all of the myofibrils inside that muscle cell. And what's going to happen now is that calcium is actually going to travel down to these myofibrils and into the sarcomeres, and it's actually going to bind to the troponin of the troponin tropomyosin complex. And as it interacts with the troponin, so you can see here calcium's binding to the troponin, what it's going to do is it's going to bind to it and cause the troponin to undergo a conformational change. And that what that troponin is going to do is that troponin is then going to pull, as it undergoes a conformational change, that tropomyosin molecule off of the myosin binding site. And that's going to expose the myosin binding site on the actin filament. So again, real quick, just to recap, calcium is going to bind to troponin. That's going to cause the troponin to undergo a conformational change. And as it undergoes that conformational change, it's going to pull the tropomyosin off of the myosin binding site on the actin, now exposing it. And now, when that happens, now the myosin heads can bind to the actin at the myosin binding site. So again, this is just showing that whole process. Another picture showing that whole process at a neuromuscular junction here. Here we have the motor neuron. It's releasing acetylcholine. That acetylcholine is now binding to the receptors on the motor end plate of the muscle cell. That when they bind that acetylcholine, that's going to cause an action potential to travel across the sarcolemma down the T-tubules. And as it travels down the T-tubules, it's going to signal the sarcoplasmic reticulum, which is all in here, surrounding those T-tubules, to release calcium. And now that calcium can go bind to the troponin of the troponin tropomyosin complex and now pull it off of the myosin binding sites on the actin. So when that happens, now myosin is free to bind to the actin. And so the myosin will actually form what's called a cross bridge with the actin. That's when it binds to the actin. That's what you're seeing here. The myosin actually binds to the actin and forms a cross bridge. Okay? And when it does that, it's going to undergo its power stroke, just like you see here. The myosin head is going to, boom, undergo this power stroke. It'll bend and pull the actin filaments. And that's what ultimately is going to cause the sarcomeres to shorten. Now, after the nervous impulse has been received, then we have an enzyme. You don't have to know the name of this enzyme, but we have this enzyme that's going to actually break down the acetylcholine so we don't continue to stimulate that muscle cell. Once that happens, then calcium will be reabsorbed. So calcium will be reabsorbed by the sarcoplasmic reticulum. Calcium will go back into the sarcoplasmic reticulum where it will be stored. And the linkages between myosin and actin will eventually be broken, starting the cycle all over again. So don't worry, I'm going to go over this again in a step-by-step process, but this just gives you kind of, again, a piece of the puzzle. And we're going to put all these pieces of the puzzle together here in just a minute. This is just another picture for you showing you the same thing. Here, an action potential has traveled down the motor neuron, cause it to release acetylcholine. Acetylcholine binds again to the motor end plate here, causing now an action potential to travel down the sarcolemma of the muscle cell, down the T-tubules, and as it travels down the T-tubules, it's basically going to signal the sarcoplasmic reticulum to release calcium. Calcium now can go bind to that troponin tropomyosin complex and pull it off of the myosin binding site. So now myosin can bind to the actin. So again, this is going to be showing you the exact same thing. So I just put in multiple pictures of this, so that way then whatever visual image works best for you, you can use. So now we're going to go through a step-by-step process, and then we're going to watch some animations on this. So again, in a relaxed state here, our cross bridges, these are our myosin heads, just to orient you. These are our myosin heads coming off of our myosin molecule. And here is the actin with in blue here is the tropomyosin, and then here in yellow is the troponin. And then here you can see they even drew in orange here, the myosin binding site on your actin. And so you can see in this case, in the relaxed state, the troponin tropomyosin complex is actually covering this myosin binding site here. So again, the myosin heads are disengaged. They cannot bind to the actin here because it's covered by the troponin tropomyosin complex. And in this case here, the myosin heads are actually cocked and ready. They're in the energized state. So the myosin heads are always ready to go, cocked and ready to go. And for this to happen, for us to see a muscle cell in a relaxed state, we have to have very little calcium in the sarcoplasm, obviously, because again, if calcium's in the sarcoplasm, then that calcium will bind to that troponin tropomyosin complex and move it off of the myosin binding site. So now we're going to see a change here, and that's what we're going to do is walk you through muscle contraction here. So now here, you're going to see one of the first things that happens again is we're going to get an action potential from that motor neuron. Acetylcholine will be released. Acetylcholine will bind again to the motor end plate. That triggers an action potential in the muscle cell that would travel across again, remember the sarcolemma, down the T-tubules and trigger the release of calcium from the sarcoplasmic reticulum. I don't care that you know the terminal cisternae, that's more Bio 201. But I do want you to know that calcium is released from the sarcoplasmic reticulum, which I just abbreviated as SR. Then that calcium will bind to the troponin. Okay, so here's again the yellow, the troponin. And when it does, that causes the troponin tropomyosin complex to undergo a conformational change. And when it does, it's going to pull it off of the myosin binding site, now exposing that myosin binding site. So now the myosin can bind to the actin at that myosin binding site. And that's exactly what's going to happen here. Here, the myosin head attaches to the active site on the actin, and it forms the cross bridge. When that happens, okay, and you're going to see there was a change here. Here, the myosin head was up. When it forms that cross bridge, that's going to cause the myosin head to undergo its power stroke. And what it's going to do is it's going to bend back this way. Okay, and that's what's going to pull those actin filaments closer together. So we call that the power stroke. That's the power stroke of the myosin head. Okay, as it ratchets that actin filament forward. So that myosin head is ratcheting. So the release of energy from ultimately the breakdown of ATP is going to cause that myosin head to ratchet. Okay, and we're going to see that the ADP, the adenosine diphosphate that had been stuck there inside the myosin molecule, is going to be released in just a minute here. And again, that's really what allows us to pivot that myosin head. That's a little more detail. I don't necessarily care that you know that happens here. But I do want you to know again that myosin head is ratcheting along the actin filament. Then, another bit of information I want you to know here is that for that myosin head to detach from the actin, it has to bind another ATP molecule. Okay? It also binds magnesium, but I want you to know that it has to bind another ATP molecule. That's very important. Once that myosin head binds another ATP molecule, then it will detach from the actin, and only then. This is important to remember because this will actually explain a process called rigor mortis to you. When an individual, without a lack of a better word to say it, expires or passes away, dies, that individual is no longer able to make ATP. And when they're no longer able to make that ATP, then there's not enough ATP to bind the myosin heads. And what ends up happening is the myosin heads stay bound, then they're stuck to the actin filament, and that holds the muscles in this contracted state, and that's what causes rigor mortis. Now eventually the rigor mortis will go away as the actin filaments over time and the myosin filaments will eventually just break down. Those will eventually break down over time. But again, this is what causes rigor mortis. So the myosin heads have to bind that ATP to be released from the actin filament. Now, what's going to happen is that as those myosin heads bind ATP and are released, then that ATP is going to be hydrolyzed, meaning the ATP is going to break down into ADP and a phosphate. And the energy, remember from that high energy phosphate bond, because we had, again, it was ATP, adenosine, triphosphate, okay? So when we broke this bond here, okay, the energy from this bond is going to be used to recock the myosin head. So now we're going to re-ratchet the myosin head the opposite direction, so that now it's cocked and ready to go for the next either muscle contraction, next signal, or if calcium's still there, then it will just continue to bind to the next myosin binding site. So that's exactly what this says. If calcium remains high, then another cross bridge will form between the myosin heads and another active site on the actin filament. However, if stimulation stops, so meaning the motor neuron is no longer stimulating that muscle cell, then the sarcoplasmic calcium levels, so the calcium levels inside the muscle cell are going to drop, and now the troponin tropomyosin complex will move back onto that myosin binding site, covering that myosin binding site again. So that is the whole process of muscle contraction. So you can see again, this is going to show you that whole process, this whole step-by-step process here. So it's just a nice, they show you, step one here again, the active site's exposed here because calcium is bound to the troponin tropomyosin complex, and it's moved it off of that myosin binding site. Here now, cross bridge is formed as the myosin head attaches to the myosin binding site on the actin filament. Here now you're going to see the power stroke as that myosin head ratchets along the actin. Here now, to be released, for the myosin head to be released, it binds an ATP molecule, and then as it binds that ATP molecule and the ATP is hydrolyzed or broken down into ADP and a phosphate, it uses that energy to recock that myosin head, and now we're ready to start all over again. So what I want to do is I want to show you again some animations here. So now I'm going to go ahead and I'm going to bring back my pointer here so I can go ahead and show you some of these animations. And I think these animations are really helpful again for you to be able to visualize what's happening here. So I'm going to take you away from the PowerPoint and show you some animations. So here's a nice animation, again, that's going to show you this mechanism of sliding filament theory. So again, to orient yourself, here are these myofibrils. Here you can see are the T-tubules, and in pink here is the sarcoplasmic reticulum all around that. So now let's zoom in here to one of our neuromuscular junctions. So here you can see acetylcholine's released from the motor neuron. It's going to cause an action potential in the muscle cell that's going to travel down the T tubules and trigger the sarcoplasmic reticulum to release calcium. That's what you're seeing here. These little black diamonds are supposed to be calcium. So now here, when that calcium is released into the sarcoplasm, it again can travel down to those myofibrils where it can actually bind to the troponin tropomyosin complex. So there's the troponin and there's the tropomyosin there. And so this is what you're going to see when I add calcium here. So when I add calcium, it binds to the troponin tropomyosin complex, moving it off of the myosin binding site and now allowing those myosin heads to bind to the actin filaments at the myosin binding site and creep along the actin filament. Okay? And you can see here, these myosin heads are, again, undergoing their power stroke, and then they'll bind another ATP, be released, and then they'll hydrolyze that ATP, recock the myosin head, and then now it can attach to another myosin binding site, allowing it literally to creep along the actin filament. So again, this is a nice animation to show you this whole process. So I'm going to show you a few more animations here as we go along, but this will be another one of the animations that I'm going to post for you on Canvas. So now we're going to go back into the PowerPoint slide, and I'm going to show you another animation on sliding filament theory here. Oh, sorry, that's the same animation. Oops, sorry about that. So again, this is just the same animation, and you can watch this animation again if it helps you. So let's go back to the PowerPoint here. Now, this PowerPoint, unfortunately, it's not working, but what it's supposed to show you is that here again are the myosin heads, and they're going to creep along the actin filaments. So here in orange are these myosin heads, and they would creep along these actin filaments. Oops. And let me go back here for 1 second. And so what, unfortunately, this animation is supposed to show you, and it doesn't show you, is that it's supposed to show you how those sarcomeres shorten and extend as the muscle cell is in its relaxed state. Well, maybe it won't go back for me here. Hold on 1 second. I'm going to escape out of this as unfortunately, my PowerPoint has gotten jammed up here. So excuse me for a second. We have these technical difficulties in recording lectures just like we do in a real classroom. So you get that real classroom feeling here with the iTunes U. So let's go back into this again. Sorry about that. And again, what this was supposed to show you, what I was hoping that, but this unfortunately animation is not working, is that as the myosin heads creep along the actin filaments, these sarcomeres will shorten. And so because these sarcomeres are, they're back to back, one right next to the other, when you get the release of calcium from the sarcoplasmic reticulum, it's going to cause all of these sarcomeres to shorten, causing the whole entire muscle cell to shorten. So again, I think I have a few more animations for you in here to show you towards the end. So we'll get back to some animations. But real quick, this is just going to talk about some of your energy sources for contraction. So again, obviously ATP is our big energy source there. And let me go back there. So ATP is our big energy source, and ATP again is in limited supply, but obviously it's very, very important for muscle contraction because the myosin heads can't be recocked and undergo their power stroke if we don't have ATP. So we actually produce ATP through our usual methods, aerobic and anaerobic respiration, cellular respiration. So again, we've got, aerobic cellular respiration produces lots of ATP, and then we have our anaerobic respiration like glycolysis and lactic acid fermentation that will also produce some ATP when oxygen is not present. So aerobic respiration is when oxygen's present, anaerobic is when it's not. And again, those are our main ways in which we're producing ATP. But this is where it gets important to understand for muscle contraction. We have this other way of restoring ATP, and that's through the substance called creatine phosphate. This is unique to muscle cells. And creatine phosphate is going to store excess energy that can be released by mitochondria, and it's present then to regenerate ATP. So creatine phosphate is going to act in a way to recharge the ATPs from ADP. So what happens is when, I'm just going to skip ahead here, when ATP during muscle contraction is broken down into adenosine phosphate and a phosphate group, okay, when it's lost that energy to the myosin heads, then creatine phosphate can actually recharge it. So creatine phosphate will actually donate its energy so that now that ADP will reform a bond with that phosphate group, forming ATP. So I want you to think of ATP as like a rechargeable battery, and the creatine phosphate is what can recharge that battery. And it's just hanging out there inside the muscle. So it's an extra way in which we can recharge that ATP. We can always make more ATP as well and we have to do that in our muscle cells. So the mitochondria can always make more ATP, but this creatine phosphate is an extra way that we can recharge those batteries. So again, whenever the supply of ATP is fairly sufficient, then we'll end up storing creatine phosphate. And then as ATP breaks down, then the creatine phosphate will transfer its energy to recharge that ATP. Now, active muscles will rapidly go through that supply of creatine phosphate, believe it or not. So then eventually the muscle cells will have to rely on our traditional sources of producing ATP from the mitochondria. And that's through, again, that process that we call cellular respiration, where we break down glucose and produce lots of ATP. Here, please don't worry about the creatine phosphokinase. I don't care that you know about that. It's getting a little too detailed for us in this course. Okay, so now we can talk about, and I'm going to go back to, if you don't mind, I'm going to go back to my pen here. Oops, I'm just going to right click here. So I can go back to my pen. Now I can draw on my slide again. Oops, and I'm going to change my pen color if you don't mind here for just a second. Let me go back, and this time I'm going to see if maybe we can see red a little bit better here. Okay, so now what's interesting is when a muscle fiber contracts, it's always going to contract to its fullest extent. This is what we call an all or none response. So when a muscle cell contracts, it is either, again, going to contract all the way, or it won't contract at all. So that's what we mean by all or none. A muscle cell cannot contract partially. So a single short contraction involving only a few motor units and therefore then a few muscle cells is referred to as what we call a muscle twitch. And so this is where we're going to kind of talk about, now we're going to build on up from a single muscle cell on up to what's happening in your, muscle fascicles. So there's a process that we call summation that occurs. And this is when a muscle fiber receives a series of stimuli, increasing in frequency, that, and I'll show you a picture of this in just a minute, reaches a point when it is unable to relax completely, and the force of that, the individual twitches will actually combine together. I'll show you this in just a minute in a process that we call summation. And again, summation, again, as you can imagine, is the word sum means addition. So this is going to be where we have these the series of stimuli that are going to add upon each other. If the sustained contraction lacks any period of relaxation in between, we'll call this tetany, or a tetanic contraction. So I'm going to show you what this looks like here. So here you can see, here is supposed to be showing your action potential that's moving. So again, if you're getting an action potential, this would be because a motor neuron has fired, okay? And so here, motor neuron is fired, causing an action potential in the muscle cell, and that causes the muscle cell to contract. That's a single twitch. Now, if a muscle fiber is re-stimulated before it has had a chance to completely relax, so here you can see it's had a chance to completely relax. And this one here in summation, that muscle cell was on its way to relaxing when it was re-stimulated. And so because it was re-stimulated before it completely relaxed, that second twitch is added on to the first one. And that's what's called summation. And this actually happens, if you want to know, you don't have to know, but one of the reasons that this happens is that here you still have calcium in the sarcoplasm when you're stimulated again here. And that releases even more calcium. So what that does is that increases the amount of tension or contractile activity of that muscle cell. Now, tetanus occurs, okay, when a muscle fiber is stimulated so rapidly, you can see that here, it's stimulated so rapidly that it doesn't have a chance to relax at all, not even a little bit, in between stimuli. So you get the maximal sustained contraction here that occurs, and this is called tetany. Now, this is not the common way. Typically, this is the common way our overall muscles contract. This is not the preferred method for muscle contraction as a whole, because what ends up happening is this causes the muscle cell to fatigue quickly. Okay, so now we're going to get into talking about multiple muscle cells and bringing on multiple motor units. So if you remember, you have to think back here to what a motor unit is. And a motor unit is a single motor neuron in all of the muscle cells that it touches or controls. So then we can increase the number of activated motor units within a muscle cell at higher intensities of stimulation. And this is what we call recruitment. So recruitment means when we bring on more motor units in more muscle cells, or we stimulate more motor units that are going to stimulate more muscle cells within an overall muscle. So as the intensity of stimulation increases, recruitment of motor neurons continues until all possible motor units in the muscle are activated. And this causes the muscle to contract with maximal tension. So what this means is, if you're, let's say, contracting your bicep muscle, okay, so that you can contract and bring your fist closer to the humerus bone, okay? So if you're doing that, you're contracting the bicep muscle, as you slowly bring that fist up, okay, you're bringing on more and more and more and more motor units, okay, until you've maximally contracted the bicep muscle. Okay, so that's what's happening as you are bringing up your hand, pulling up your hands. Okay, as you're doing that process, you're bringing on more and more and more motor units. Okay, that are increasing again the tension inside that muscle until you've eventually reached your maximal tension in your muscle. So hopefully that helps you to understand what's happening in the overall muscle, as that's called motor unit recruitment. Now, summation and recruitment together. So again, summation happening in individual muscle cells, and then recruitment happening again as we bring on more and more and more muscle cells, can produce a sustained contraction of increasing strength. Muscle tone is achieved by, so if we talk about, a certain muscle tone, and we see this especially in our muscles that are involved with maintaining posture. These muscles are in a continuous state of sustained contraction of the motor units within that muscle. So those motor units are continually stimulated, and those motor units are continually, again, stimulated, and that muscle is in a continual state of contraction. And that's because those are muscles, these are special muscles that are going to be associated with things like posture. So they have to maintain a special muscle tone. Okay, so again, and we're going to stay, that's as basic as we're going to stay. We're not going to get into any more nitty-gritties on that, because that is, making muscle is probably more complicated than you ever imagined. And so again, I'm sure I'm going to get questions this week, so please feel free to email me as you have questions on this. So again, that was talking about our basics on how muscle contraction works in skeletal muscle. Now we're going to talk a little bit about how smooth muscle works and cardiac muscle. Don't worry, this is very simple because it's very similar to how skeletal muscle contracts. So when we look at our smooth muscle fibers, our smooth muscle cells are going to be elongated with these tapered ends. So they're going to look very different than our skeletal muscle. And they're going to lack striations. So they don't have these true sarcomeres that create the striations that you would see in a skeletal muscle. And they have these really large nuclei as well in the center of them. That's one of the ways that we can identify smooth muscle here, is that you're going to see, again, you have these tapered cells, and they're going to have these really large nuclei. Okay, and then no striations. So that's a way that you can identify, and you, hint, could always get a picture on an exam of muscle, and I could ask you, what type of muscle is this? You would be able to tell that this is smooth muscle because they have these big, large nuclei, and you don't see any striations. It's the only muscle that you're not going to see striations in. Honestly, I'm not going to expect you to know multi-unit. I should have cut this slide and I forgot to, so don't worry about knowing the difference. This is more Bio 201 material. You do not have to know the difference between multi-unit and visceral smooth muscle. Although, just so you know, visceral smooth muscle, it tells you one of the main places that you can find smooth muscle, and that is that you're going to find smooth muscle in the walls of your hollow organs. So all throughout your digestive tract, all through your gut tract, you're going to have smooth muscle, and that smooth muscle contracts to form these peristaltic contractions that moves food down your digestive tract. So the smooth muscle is responsible for that. Smooth muscle, again, is going to contract in a very similar manner that skeletal muscle is going to contract. So the myosin is going to bind to actin. It's just that it's in the myosin and the actin look a little bit different because they don't form these true sarcomeres. You're going to see here would be the actin filaments in blue, and here are the myosin filaments in pink. And again, the myosin filaments are still going to creep along the actin filaments. It's just that they're going to do it, again, in a different direction or different format. So here you can see this is showing the, relaxed state of the muscle. And here in the contracted state, you can see the myosin and actin overlap quite a bit here. It just forms, the myosin and actin form this more net-like arrangement throughout the smooth muscle. So again, in smooth muscle contraction, you're going to see also that acetylcholine is not the only neurotransmitter, but norepinephrine is also another neurotransmitter that can stimulate muscle contraction in smooth muscle. So that's all. Again, that's the big thing out of here is that acetylcholine as well as norepinephrine. There's two different types. In skeletal muscle, it's only acetylcholine, but in smooth muscle, you can also get stimulation from norepinephrine. Hormones can also stimulate or inhibit contraction in your smooth muscle as well. And smooth muscle is slower to contract and slower to relax than skeletal muscle, but it can contract for a longer period of time using the same amount of ATP. Okay, so it's very efficient. So again, it may be slower to contract and slower to relax, but it can contract longer and it uses the same amount of ATP. Okay, cardiac muscle. When we look at cardiac muscle is going to look a little bit different as well. It's going to have striations like skeletal muscle, but it's going to have these things called intercalated discs. Okay? And these intercalated discs are going to allow things to move from one cardiac muscle cell to another. Also, it's going to help to hold the cardiac muscle cells together. So for example, this is one cardiac muscle cell here, and this is a cardiac muscle cell here. And through these, what we call intercalated discs, we're going to have little holes in them called gap junctions. And that's what's going to allow calcium from one cell to move into another cell. So what that does is as calcium again is released in one cell, that calcium that can move through these gap junctions into this cell and into this cell, into the neighboring cells. And what this does is this allows all of the cardiac muscle cells to contract together as one. And this is important because, of course, cardiac muscle is in your heart. And so we need all of those cardiac muscle cells to contract together so that it contracts as one unit. So the heart will contract as one unit. The other thing that you're going to see in cardiac muscle is that cardiac muscle has these things called transverse tubules, just like skeletal muscles, but those transverse tubules are going to store some extra calcium in them. And this will actually allow a cardiac muscle cell to contract for a longer period of time as well. So again, I already kind of introduced this, but again, the cardiac muscle cell contains these intercalated discs. They're just these discs that join one cardiac cell to another cell and allow it to transmit the force of contraction from one cell to the next. So again, this allows us to rapidly transmit that impulse throughout the heart so that all of the heart cells will contract at the same time as one. The other thing that you probably already know about cardiac muscle is that cardiac muscle cell is self-exciting and what we call autorhythmic. So again, the cardiac muscle cells through what we call the SA node, we'll get to this when we get to cardiac. And this isn't as important that you understand this now because this is going to be coming up here very soon when we cover the heart. And we'll talk about how the cardiac muscle cell is autorhythmic. Now real quick, I'm going to escape out here for just 1 second because I want to go back to the very beginning here. I'll remember what slide I'm on, slide 56. And I want to go back to the very beginning here and just show you a picture of the different types of muscle. So here, now hopefully you'll recognize this. Oops. You'll recognize this as skeletal muscle. It's striated and the cells have many nuclei. So you can see that. Striated, the cells are elongated and they have many nuclei. So that's skeletal muscle. This here is going to be smooth muscle because it's not striated. The cells are tapered and they have really large nuclei. And here is going to be your cardiac muscle. And again, you're going to see in your cardiac muscle cells that you can see. It's hard to see in this picture, but if you were to look at an actual microscope image of it, you would see the striations a little bit better. And the big thing is, in this one you can see these are the intercalated discs. Here's an intercalated disc here, here, here. These are the intercalated discs. So I just wanted you to be able to see some actual microscope images of these three different types of muscle: skeletal muscle, smooth muscle, and cardiac. Okay, so now let's jump back to slide 56 here. Don't worry, we don't have much left. And keep in mind that this is actually 2 lectures in one. So you're actually getting 2 lectures in one here. I decided not to separate it because it would be silly to separate it here. Okay. So now this last bit of the lecture is going to talk a little bit about some of your skeletal muscle actions. And so this will be hopefully useful in understanding some of the actions of the different muscles. So when we talk about skeletal muscle actions, we have to talk about the origin and insertion of a muscle. And oops, I'm going to go ahead and bring back my pen here. So when we talk about the origin and insertion for muscles, the origin is going to be the immovable end of a muscle, while the insertion is going to be the movable end, or the end that the bone that's going to be moved. So for example, let me just give you an example of the biceps muscle. When you talk about the bicep muscle, the origin of the biceps, you actually have 2 points of origin, and they're both on the scapula. So that is going to be the end that is not going to move. The scapula is not going to move. Okay? So that's the origin. The insertion of the biceps is going to be the radius on the forearm. And that's going to be because as that muscle contracts, the scapula is not going to move, but the radius, okay, is going to move. As that muscle, the bicep muscle, contracts and it flexes, okay, the forearm, it's going to pull that radius, okay, closer towards the humerus. So that gives you an example of origin and insertion. Now again, as you can see here, some muscles have more than one insertion or origin, and the bicep muscles are an example of that. The bicep muscles are actually called the biceps brachii, and that's because the bicep portion, bi means 2, so it means that it has 2 origins. So, and they're both on the scapula, just in different places on the scapula. So that just gives you an example of that. Now, when we talk about the interaction of skeletal muscles, we have what's called the prime mover, and this is the muscle that's doing the majority of the work. And in this case, I'm going to use the biceps again. When we talk about flexing the forearm, the biceps brachii would be your prime mover. Okay, just as an example. Okay, so they're the one that are doing, the bicep is doing the majority of the work to flex our forearm. Okay, now helper muscles are called synergists. So we have a muscle that lies right underneath the biceps brachii called the brachialis, and it's going to help flex. It's not the primary flexor, but it's going to help the biceps brachii flex the forearm. And then lastly, we have opposing muscles that are called antagonist muscles. And they're going to be the muscles that are going to do the opposite action. So in this case, if our biceps are flexing the forearm, then our antagonist muscle would extend the forearm. And our antagonist muscle would be the example of this would be the triceps in this case. So again, here this is just showing you, again, our biceps would be our prime mover if we're talking about the action of flexing the forearm. And our triceps would be the antagonist. Now, if we were talking about the action of extending the forearm, then the triceps would become the prime mover and the biceps would be the antagonist. So it depends on what action you're doing. Okay, so again, if you're flexing the forearm, then the biceps would be the prime mover. If you're extending the forearm, then the triceps would be the prime mover. Okay, now when we talk about skeletal muscles, and because you have to know some of these for lab, I'm going to help you out in terms of how we name muscles. So muscles are actually named according to a number of different criteria, and we can use any number of these. So first off, muscles can be named after their size. So for example, the pectoralis major muscle is one of your major chest muscles, and it's named after the fact that it's a very large muscle, hence the name major. We can also name a muscle based on its shape. So something like the trapezius muscle is shaped like a trapezoid. Okay. So that's your trapezius muscle has a trapezoid shape. We can also name a muscle based on its location. Subscapularis, as you can guess, is located below the scapula. We can also name a muscle based on its action. Levator scapulae, as you can guess, that is a muscle that elevates the scapula. We can also name it based on the number of attachments. So biceps brachii, bi means two. So again, that's a muscle with two origins. We can also name it based on, again, its insertion as well. Direction of the muscle fibers. So the external oblique muscles, the external oblique muscle, it runs on your abdomen, and oblique tells you that the muscle fibers run in an oblique fashion across. So the rest of, you'll be happy to know that the rest of this lecture, these pictures, are just to help you out with lab. So you can use these pictures, or if you don't want to use them, you don't have to, but these are for lab. You will not be required to know certain muscles, and I won't show you a picture of a muscle and ask you what that muscle is. You will not be required to know that for lecture. That's only for lab. So all of these are just to help you out with lab. All right, so that concludes the lecture. I know that this lecture is very involved, so please feel free to email me with any questions that you have, or again, please write them on the discussion board so that everyone, and actually that's always a better option, so that everyone in the class has the opportunity to read that question and answer. All right, have a good night.